Hi, this is Judah with Awesome Science. And today I'm going to be discussing with you about how the cell cycle is controlled by internal and external mechanisms and how loss of this control can lead to cancer. As we should all know, eukaryotic cells are cells inside our body that contain a nucleus and also known as animal cells. These cells move through the cell cycle through a regulated way, meaning they use information about the state of the inside and outside of the cell, including the environment around it. Regulation makes sure nothing goes wrong when the cells divide. However, non-normal cells, such as cancer cells, move too fast through the phases, causing a disruption of the cycle. In the cell cycle, there are three checkpoints that a cell must go through, the very important checkpoints, known as G1, G2, and M. The first checkpoint, G1, is one of the most important because it decides whether or not the cell can pass into division. This checkpoint checks for cell size, nutrients, growth factors, DNA damage. Once the cell passes through the G1 phase, it will continue to the S phase, and then the rest of the cell cycle where it is committed to the cell division, meaning it cannot go back. The next checkpoint is G2, also known as the M phase. That checks if any DNA has been damaged and if the DNA has been completely copied into the S phase for cell division to go smoothly, as we can see in the G2 checks over here. If any damages are found, this cell will pause at this stage until it is repaired. If the damage is irreplaceable, then the cell goes through a self-destruction mechanism to make sure the damaged DNA does not pass on to the daughter cells. This is a key part in preventing cancer because cancer cells ignore the signs telling them not to divide, meaning that they can pass on damaged cells without self-destruction. The third stage is the S spindle stage, or the M checkpoint. At this stage, it checks for chromosome attachment to spindle at a metaphase plate. The cycle cannot proceed until all chromosomes are firmly attached to the spindles, as you can see over here. The checkpoint works because when a chromosome is misplaced, the cell will pause mitosis, allowing time for the spindle to capture stray chromosomes. Two very important processes in the cell cycle are cyclin and cyclin-dependent kinases, also known as CDK. CDK, as you know, is inactive until it is joined by cyclin, which activates it. As you can see here, it's joined by the G1 and S cyclin. Cyclins drive events of the cell by partnering with the CDK, as you can see over here, which are enzymes breaking down the cell easier or faster, as we know enzymes to do. This may seem confusing, so I'll break it down a little. CDKs, or the cyclin-dependent kinases, are mixed with the cyclin and direct specific target proteins controlled by the cyclin, right here. Then these proteins go to the S phase, begins, and the enzymes are activated, meaning that it starts the breakdown of the nuclear membrane. Another important part of the cell cycle, or a good example of how cell cyclin and CDK work together, is through MPF, also known as maturation promoting factor. The MPF makes M-glycine more complex by adding phosphates to the targets, the protein targets, inside the nuclear membrane, causing a breakdown of the nuclear membrane by promoting chromosome condensation, as you can see right here. Another important aspect is PDGF, which is a family of molecules released from tiny parts of the cell found in blood, known as platelets, which is also known for use in blood clots. Forms of PDGF help heal wounds, repair damage to blood vessel walls, and they also help blood vessels grow. And it's important to know that if the cell cycle loses control, that it can cause cancer, but we want to know how it leads to cancer. As you can see in the first diagram, the normal cells need positive growth factors for the cells to divide. And when they are given negative growth factors, there is no cell division. But this is not the case for the cancer cells. As you can see, it doesn't matter if it's positive or negative growth factors, the cells divide anyway. This is because the cancer cells go through the cell cycle disregarding signals telling them to stop dividing, causing them to multiply at a rapid rate, bringing along damaged DNA and other harmful factors. Cell cycle regulators can also cause cancer. A positive cell regulator may be overactive, as in sending signals when growth factors are not there. Negative regulators can also become inactivated. For instance, a protein that stops the cell cycle from continuing because of DNA damage may no longer sense damage, or it might not trigger a response. This is the end of the video. I hope you learned some valuable information. Thank you for watching.